Now, you've heard about science fact this morning, and now what I want to do is talk about some science fiction, because really what I want to do is try and tell you maybe where we were going in the future. Now, obviously what's new in the lab is an issue, and some of it I can't tell you about, because if I tell you about it, it probably won't become a treatment of tomorrow, because what we need to do is make sure we protect all the information we have so we can properly develop it. So the ideas that I have today may be a treatment for tomorrow, but in reality, it's not really a treatment of tomorrow. And what we need to get across is it takes time, and it may be 10 to 15 years. Now, we run a blog, and this week we had this thing, and he said, if I was Professor Giovannoni, I'd tell Professor Mouse that he's got 3 years to come up with the goods, get a cure, or have him sign, go down the benefits office. Now, first thing for you is maybe I have got the cure, but if I have the idea today and get a drug, it's a long process. So we need to make a drug, and then allows us to get onto this board game, and this is like a Monopoly board, and the more you go round, the more expensive it becomes. And we have a new drug. We have to make sure it's safe in animals first, then it's safe in humans in the phase 1 trial, that it's safe in people with MS, and then we have to show it works in a phase 3 trial. But that's actually quite a simple thing, and actually it's not just a question of showing it's safe in humans and then carrying on. You have to go back and actually show it's safe in animals for a bit longer, and then when you do the phase 3 trial, you've got to do 2 of them. And actually, for a drug company to actually get the drug all the way around the board game costs about $1.5 billion. And it takes a drug company 10 to 14 years to actually do that. And as a researcher, we don't have the resource. So we can probably get around the board for about 50 times cheaper, but it'll take us a bit longer. So, if I do have the cure, it's not going to become as quickly as we all wanted it. Now, the question I'm going to try and ask is progression. Why don't we have any drugs? And I think one of the issues there is time. So it was only probably about 15 years ago that we realized that actually just dealing with the immune system wasn't going to be enough for people with progressive MS, and what we need is probably a different type of treatment. Now, it's not we don't have any ideas. We've got lots of ideas, and the international community, led by the MS societies, are really focusing on progressive MS. Now, there are a number of reasons why you don't have drugs, but one of the reasons I want to focus on is side effects. Because if you take a drug and you have a side effect, you may not want to take your drug because it's causing problems. And if you don't take your drug, you won't get an effective dose, and the drug won't work. And actually, that probably has happened for quite recently with one trial where about 50% of the people weren't taking the drug. So how can a drug work if people don't take it? So what we've been thinking about, well, is how do we avoid the side effect issue? And one way is to deliver drugs where they're needed. So this is an MS lesion, and what you can see is those blue dots. That's the MS lesion, the white blood cells, surrounded by a blood vessel. So one of the thoughts, as Professor Miller showed you, is you stop those white blood cells arriving in the brain, and that will be advantageous. But for people with progressive MS, we have to deal with the damage that occurs as a consequence of these cells coming into the brain, and that means we have to get our drugs into the brain. Now, how do we get drugs into the brain? Well, the brain has evolved a system which tries and excludes things from the blood getting in. And so if I have a drug, which is that little thing in the top left-hand corner, it can have its effect in the blood and maybe knock out white blood cells, and that's how most MS drugs currently work. But to get into the brain, they have to get through the blood-brain barrier, and one of the parts of this blood-brain barrier is it's a wall of fat. And the cell membrane of the blood vessels are fatty. And I think you'll all know that water and fat don't mix. So if you have a drug that is soluble in water, it'll be difficult for it to get in the brain. So you could just make your drug a bit more fat-soluble. But the brain's also got things that can still deal with that. And there's some pumps, and one of these pumps is called permeability glycoprotein, called P-glycoprotein. It looks like a peg. And what happens with this pump is it sucks in potentially fat-binding chemicals into the hole of the peg, and with a bit of energy, it opens and throws the drug straight back out. And so that stops many drugs getting into the brain. Now, this is the simple way. This is actually what it looks like in reality. But the brain has many different pegs. It maybe has about 30 different pegs. Now, I've told you this story. What is the significance of that? Well, when we look in the brain of an animal with an MS-like disease or somebody with MS, what we notice is we can see the P-glycoprotein, the drug pump, in brown. And what you'll notice in the middle, where all those blue cells are, is that there is no brown because the pump has disappeared. And that's interesting. Now, if we look at this one, this is a different pump, but you can see this pump expressed on the blood vessel surrounded by these white blood cells, whereas this one, you can't see any brown, but the blood vessel is there because this pump has disappeared. So if we can target a drug to that drug pump and it's not present in the MS lesions, it means the drug can get into the brain. So that's actually what we try to do. So here's a graph. What you do is you take your pill, you then get levels of drug into the blood. If it was a brain-penetrant drug, you'd get brain levels of the drug. Now, that could cause side effects if the drug is targeting something that's important. So what we tried to do was make this drug here, the new drug, and it's targeted to the pump. And so what happens is, when we give the drug, we can find it in the blood, but it doesn't get into the brain. So what we then hope is that in the MS condition, you'll get small amounts of the drug into the brain, into areas where it's needed. Now, that's this hypothetical. But what we actually saw is we still couldn't actually detect this drug in the brain. And that's because this drug is really like the prototype. It doesn't give us the nice profile that we'd like to see with the big solid red line. So it's like an early drug. But what happened was, if you were a chemist, you'd see this bit and you go, well, if you shine a UV light on that drug, it'll glow in the dark. And that's exactly what we could see. So this is the brain of something where we've got an MS-like disease, and you can see the green drug getting it into the brain. So although we couldn't pick it up easily, we could actually physically see it in the brain. And if we stain cells blue, you can see here that where the lesions are is where the drug is getting. And so you can see that the drug is getting exactly to the area where we want it to go. So what's the consequence of that? So if we look in a healthy mouse, or you could think about a healthy human, that you've got a certain level of nerve content. And what we know with progressive MS is that nerves are damaged, and so you will lose nerves as a consequence of your MS. Now, if we stop the white blood cells getting in, then of course there would be no damage, there would be no nerve loss. But in terms of thinking about progressive disease, disease has occurred and these drugs don't necessarily work as well. So this drug doesn't stop the disease occurring, but what it does do is it stops the nerve loss as a consequence of disease. Now, will this be developed as a drug for multiple sclerosis, or will it be put into the bin of scientific failures? And that largely depends on some of our clinical colleagues. And what they're doing is doing clinical trials in multiple sclerosis with a drug that binds to the same target as our new drug, but these ones get into the brain without having this specific targeting. And you may hear later on about this optic neuritis trial with a drug called phenytoin, which is fully recruited. And there's another trial called the Proximus trial, which is starting to recruit. So if that works and gives us an indication that this is a way forward, we can then think about how to be smarter with the drugs to avoid the side effects that some of these drugs invariably may have. So that's an idea, and it would be 15 years away from being delivered to humans. So what I want to do is talk about some other stuff that we have done in the lab, and that's in terms of symptom control. So if your brain says, I want to move, it sends a signal down the spinal cord and into the muscle saying move, and then the muscle sends a signal back up to the brain saying, I've moved. Now, in multiple sclerosis, we know the problem is in the brain and the spinal cord, and what can happen is that you get too much signaling, too much excitation, and that causes spasticity, which is the limb stiffness. So there are drugs, which you'll be aware of, that can have some effect, such as baclofen. These get into the central nervous system, but potentially they will cause side effects because they can get into the brain, causing, sedation. So we had the idea, could we interfere with the exaggerated signaling outside the brain and spinal cord, and then target that towards the nerves in the periphery rather than in the spinal cord of the brain? And how could we do that? Well, I've shown you. We just made the drug very soluble in water, and so it doesn't really get in the brain. And this drug is called VSN16, which I've talked to you before. It's largely seen excluded. And so, the government wants us to talk about animal experiments. So what I'm going to show you is what happens to a mouse when you give this type of drug. And what you do is you get muscle relaxation. So the drug works just like baclofen would, but the problem with baclofen is its side effects. It can cause sedation, it can make you floppy, it can make you, give you cog fog. What we found with our drug is because it's not getting in the brain, it's not having any side effects. Now, where are we in terms of the development? So we started with this drug in 2000. We got onto the board in 2005. And I came to talk to you, or some of you, in 2011 about this. And where are we now? Well, we finished our animal safety studies in October of 2013. We started our human safety studies in December of 2013. And I'm pleased to say that last week we finished the studies in humans. The drug is safe, and as far as we see, has no side effects. So the next stage is obviously to try this in multiple sclerosis to see if it's safe and whether it works, and that will depend on whether we can get enough money to do this. Now, what the important factor here is we started in 2005, and we'd be in 2015 before we know if this drug works. So it will take us at least 10 years to get that around this part of the board, and we still have to show it really works properly in multiple sclerosis. Now, you'll also notice we started in 2002, and we're now in 2014. That's 12 years from my idea to almost just getting it into humans. So it takes a very, very long time. And that's what we need to get over as scientists, that things aren't a quick fix. But you can speed the process up. So what you can do is you take a drug that's been all the way around the board game and is known to be safe in humans, and then you move that into multiple sclerosis, allowing you to quickly go to the phase 2 or even straight into the phase 3 to slow this process or speed this process up. And this is what the pharmaceutical companies have done for most drugs that are available as we speak. Now, in terms of what's happening in the lab, I can say we and others have identified many different currently available drugs that could potentially affect neurodegeneration. In our lab alone, we've got 10 different candidates. And this year, there's a number of labs have also found drugs which are safe in humans, which could be useful in repair. So the big question is can neurologists deliver the drugs to you? The answer is we'll have to wait and see. And if you come to our blog, which is here, we'll tell you the answer as they do it. So thank you.